Welcome back, Brookings Bio students. We're going to keep going on our learning about cell parts. In this video, last video, we talked about the plasma membrane, the cell membrane, and this time we're going to learn about a couple new cell parts. We're going to start with nucleus, and nucleus is one of the largest organelles, easy to see when you look at a cell under a microscope. It's one of the biggest cell parts, and if you look closely at a nucleus, you'll see that it's surrounded by another double membrane called the nuclear envelope, also sometimes called nuclear membrane. But envelope is something that holds your letter, and our nuclear envelope is going to hold our DNA, our information. If you look closely now, remember we talked about that bilayer in a plasma membrane and a cell membrane. The phospholipids line up like a sandwich with the heads toward the outside and tails towards the middle, those phobic tails. They don't like water. And notice now we have two bilayers, not only a double layer here, but two bilayers, a double layer surrounding our nucleus. And in order for things to move in and out of the nucleus now, we need to have some openings. Remember, our DNA is our information molecule that carries the code, but it stays in the nucleus in a eukaryotic cell. And RNA is our transfer molecule. RNA is going to carry the information from the nucleus out to the cell, and it needs a way to get out. And nuclear pores are little openings that allow molecules to move in and out of our nucleus. Thing about pores, P-O-R-E now, it means a little opening, not a P-O-U-R like pouring water, pouring a liquid. Look at your skin. You have little pores in your skin, little openings. And these openings allow things to move in and out of our nucleus through that nuclear envelope. Inside now we're going to have our genetic material, DNA. And DNA can exist in two forms depending on what the cell is doing. In both cases, the DNA molecule, the DNA information stays the same. What's different is how tightly we pack that information. If you remember in middle school, you learned about mitosis, cell division. And when I'm moving my chromosomes around, I don't want to leave anything behind. I don't want it to get broken or torn. And so packing it really tightly is a good idea. It makes it easy to move. And so chromosomes is our tightly packed, scrunched up DNA, easy to move, and we see it in cells that are dividing, mitotic cells. Most of the time, though, the cell needs to have the information available. The DNA needs to be open so we can read it. And so DNA that is less tightly packed, it's still coiled up, but it's not as tightly packed as a chromosome. That form of DNA is called chromatin. And the cell can switch back and forth between those two forms depending on what it's doing. Chromatin, the DNA is open and spread out like a book. Open the book so we can read it. And we see that in cells that are not dividing, when the cell is using the information to do its job. When we want to move the DNA around, we scrunch it tightly into chromosomes. Think about when you take your homework home. When you want to move your books and your notebooks, you pack it tightly, close it up, we put it in our backpack, and easy to transport. You can carry it home, but when you want to work with the information, when you want to use it, you have to take the books out, open them up, read your homework assignment, use the information to do your homework, and then when you want to carry it back to school, what do? You close the books, you close your notebooks, you put them back in the backpack. Makes it, if you close them up, it's easier to transport. And that's how the chromatin thing works. That cell is going to unwind or tightly pack its DNA depending on what it's doing. DNA now is going to be our genetic code molecule, and it has the information to tell all the other cell parts what to do. So we call the nucleus the control center of the cell. If I look closely at a nucleus under a microscope, I see this little dark spot here, And it's because there's a lot of chemical activity going on there. It looks dark. And that little dark spot is called the nucleolus. Notice now the words look a lot like, that word looks a lot like nucleus, has some of the same letters. What I got to look for and help me remember is that O, nucleolus. That's where we're doing a lot of chemistry, and we're making the RNA that we need to build the ribosomes. We're going to talk more about ribosomes in our coming slideshows, but ribosomes are cell parts that are going to take the information from the DNA and use it to make proteins that do something in our cell. And so there's a lot of activity being made. The RNA for ribosomes is made here, and that's why it looks a little darker than some of the other places in the cell. Okay, so let's get some information in our Flipbook. Find your Flipbook, get out there, find the nucleolus page, and surrounded by a double membrane. Now, it's one of the few cell parts that has a double membrane around it. We're going to talk about a couple more in the coming videos. Called the nuclear envelope. And remember, our nucleus now is the control center of our cell. It's going to hold the genetic material that tells all the other cell parts what to do. And we have pores, little openings that allow molecules to move in and out, like the RNA that's carrying the information out to the cell, to be able to get out of our nucleus. And nucleus now is where we're going to store our genetic material, our DNA. The nucleus, if you look closely, and you can see it here in the little diagram, that little dark spot in the nucleus where we're doing ribosomes, making ribosomes, is called the look at that nucleolus. And its job is to make the RNA that builds the ribosomes. It's part of the ribosome building block that makes up the ribosomes. I want you to do this, because remember, anytime we can do something to help us remember a cell part or remember a vocab word, that's a good thing. So I want you to take a dark color, and in your Flipbook, I want you to fill in this little O, because that makes it kind of stand out, and it helps me remember what it looks like. Remember, the nucleolus, olus is a dark spot in the nucleus. So we're going to fill in and make a little dark spot in our word. And also helps it stand out so I can see, remind me that when I see that O in that word, it's not nucleus. It's talking about nucleus, nucleolus, or also called nucleolus sometimes. I had a teacher that used to call it nucleolus. Okay, so let's look at our DNA now. That's where we're going to have our DNA in our cell. And DNA can be in the two different forms depending on what it's doing. Still the same DNA molecule. But if we scrunch it up tightly, we call it a chromosome. And if we unwind it slightly and less tightly packed, we call it a chromatin. Chromatin, chromosomes. And the DNA can switch back and forth between those two forms depending on if they're dividing, tightly scrunched, or non-dividing, open so we can use the information. Okay, we're going to stop right here for a second. We're going to go to our cell picture, and we're going to see if we can color in our nucleus. So I'm going to ask you, last time we colored in our cell membrane, our plasma membrane, so we're going to pick some new colors now. Remember, we're kind of color coding this, so I want you to find a new color that you haven't used before. And I'm going to pick purple. Oh, let's try different. Let's try this. I'm going to color in. Now here's my, notice it's been kind of sliced so you can see inside. Looks kind of like an orange that's been sliced, but you can see the membrane here around the outside. Don't color the little holes yet. Those are openings to let things in and out. We're going to color the membrane that's around my nucleus. And notice animal cells have it. And plant cells, look over here. There it is again. Don't color your holes. We're going to color our nuclear envelope. The membrane that goes around. Remember, it's a double membrane around my nucleus. And now if I look in my bacteria cell, I don't see that cell part. And that's because bacteria, remember the definition of a prokaryote is a cell that does not have a nucleus. It has DNA, but it's not contained in a nuclear envelope. So if you look at your label now, notice nucleus is only under eukaryotes. So I'm going to underline nuclear envelope, or we can color it in the letters. Color coding, remember, that's my nuclear envelope color. And then I'm going to ask you to pick a new color to be your nuclear pore color. And we're going to color in our little openings that allow things like RNA to move in and out of the nucleus. DNA is going to stay in the nucleus where it's protected. But those nuclear pores now allow things to move in and out. Carry the information from the DNA out to the cell. Remember, that was one of our functions of RNA in our first chapter. Okay, I'm going to ask you to pick another new color now. Everything's color-coded. And we're going to color the DNA. It looks kind of like spaghetti. All this kind of spaghetti looks like little worms inside. This is our DNA molecule. And remember, it's stored in the nucleus in a eukaryotic cell. DNA is in the nucleus. And depending on whether the cell is dividing or non-dividing. So look at your DNA. It's kind of all loopy and open and spread out. This would be the chromatin form. Cell is working, doing its job. And now notice, bacteria didn't have a nucleus, a nuclear envelope, but it does have DNA. This little loop here is our DNA in our bacterial cell. There's a single chromosome, one chromosome. We have lots of chromosomes in our cell. You have 46 chromosomes in your body cells. And they're linear, they're in straight lines. But bacteria DNA is a little bit differently. Their DNA is in a loop, a circular piece of DNA, and they only have one chromosome, but still DNA. Remember, shared universal genetic code. We have one last thing to color, and that is our nucleolus. I'm going to ask you to pick a dark color because that helps you remember. Dark spot in our nucleus. So here's our little dark spot. Remember, that's where we're making the RNA for ribosomes. I'm going to talk about that cell part in a little bit. Nucleolus. I'm going to fill in my label here. Fill in the O. Plus. I think we're done. We're going to talk about some more cell parts in the videos to come. Thanks for listening.